All right, let's talk about something we literally walk all over every single day: dirt. We're going to go on a journey from, well, dirt, all the way to life-saving drugs. It's a secret history, really, all about a hidden war that's happening right under our feet. So let's dig in.
So let me ask you this. Just for a second, forget everything you think you know about dirt. What if the ground you're standing on right now wasn't just ground? What if it was a bustling, microscopic city, a world packed with conflict and alliances, holding secrets that could literally change the course of human history? For most of human history, that's exactly how we thought of it. Soil was just dirt. It was lifeless, inert stuff you put seeds in. It wasn't the star of the show, not by a long shot. It was just the stage. And for centuries, nobody really questioned that. But the truth? The reality is absolutely mind-blowing. We now know that in just a single handful of healthy soil, there are more living things than there are people on the entire planet. Billions of them. So it's not a stage at all. It's a hidden universe, a whole ecosystem that we're really just starting to explore. So how did we get our first peek into this world? Well, it was through some pretty simple but honestly brilliant techniques. Back in the 1930s, scientists came up with something called the buried slide method. And it's just what it sounds like. They'd stick a glass slide into the dirt, leave it there for a bit, and then pull it out to see what had started growing on it. It was basically like sending the first ever camera down into that microscopic city. Okay, so one of the first huge mysteries that this new science started to unravel was a question that's, kind of crucial for our survival. What actually makes soil fertile? How do plants get the food they need to grow? For centuries, farmers knew this trick. They knew that certain plants, like clover or beans, seemed to magically make the soil richer. If you planted a crop after them, it would just flourish. And scientists, like this guy J.B. Boussingault, they proved it was real, but they had no idea how it worked. It was like these plants were literally pulling fertilizer straight out of the air. And guess what? It turns out they pretty much were. Just look at this timeline. It really shows you how long it took to crack this puzzle. So in 1838, Boussingault confirms that, legumes are fixing nitrogen. But then, radio silence for almost 50 years. It wasn't until 1886 that a couple of scientists figured out the real culprits: tiny bacteria living inside the plant's roots. And then, a few years later, a guy named Winogradsky shows that some bacteria can do it all by themselves, no plant needed. The secret was finally out. The real master chemist of the soil? They're the microbes. And that name, Winogradsky, is a really big deal. This guy, Sergei Winogradsky, is basically considered the father of soil microbiology. And it's because he didn't just find new things. He completely changed how we thought about life on this planet. So Winogradsky's work uncovered this totally new way of living. I mean, we all learn about photosynthesis in school, right? Plants use sunlight for food. Simple. But what he found were bacteria that could, for all intents and purposes, eat rocks. They lived off of inorganic stuff like sulfur or ammonia. He gave them the name autotrophs, and this was a huge deal. It was proof that a whole complex web of life could exist deep underground with zero sunlight. And this thing right here, this is his most famous invention, the Winogradsky column. And I know, it looks super simple, right? It's just a glass tube filled with mud, water, and some interesting nutrients like shredded newspaper and egg yolk. But that's the genius of it. This simple column becomes its own little world, a miniature ecosystem that perfectly mimics all the different layers you'd find in real soil. All they had to do was add a light source, and then he could just sit back and watch these microbes sort themselves out. Over time, you see these amazing colored bands start to form, and each band is a totally different neighborhood of bacteria. Down at the bottom, you've got bacteria making this smelly gas, and that gas becomes food for the green and purple guys in the layers above. And then even higher up, another group finds its own perfect spot. It's literally a living, breathing model of that layered city under our feet. So understanding that there's this whole world of microbes in the soil, it leads to a new question. If all these tiny things are competing for space and food, what kind of weapons are they using against each other? The answer, as it turns out, wouldn't come from some carefully planned experiment. Nope. It came from one of the most famous lucky accidents in the history of science. The year is 1928. A scientist, Alexander Fleming, gets back from vacation and finds a mess. One of his petri dishes is contaminated with a common mold. He could have just thrown it out, but he looked closer and he noticed something incredible. Right around the blob of mold, there was this perfect clear circle, a kill zone where the dangerous staph bacteria just couldn't survive. That mold was producing something that was wiping out a major human enemy.
So Fleming figured out the mold was from the penicillium family, so he called the mystery substance penicillin. But, and this is a huge but, he just couldn't make enough of it, he couldn't isolate it, couldn't produce it in any useful amount. So he published his research, and for the next 10 years, nothing. This world-changing discovery just sat there, gathering dust in a journal. The key to a medical revolution was found, but it was basically locked in a box. Okay, so if Fleming's discovery was all about a lucky break, the next part of our story is the exact opposite. This was no accident. This was a deliberate, systematic hunt. The goal was to literally go mining in the soil for those microbial weapons. And this really shows you the two totally different ways science can work. You have Alexander Fleming on one side, with his once-in-a-lifetime accident, and on the other, you have Selman Waksman. He was a soil microbiologist, and he had a powerful idea. He thought, hey, if all these microbes in the soil are constantly at war, the dirt must be full of their chemical weapons. So he decided to go find them, on purpose. And boy, was he right. Waksman's lab basically turned into an antibiotic factory. He's actually the guy who came up with the word antibiotic. His team methodically screened thousands and thousands of microbes from the soil and ended up discovering more than 20 new antibiotics. This was it. This was the birth of the way we look for new drugs even today, a systematic hunt for nature's secrets. But the absolute crown jewel of their work was a drug called streptomycin. Now, you have to understand, at the time, tuberculosis was a monster. It was a global plague, and getting it was basically a death sentence. But streptomycin, which they got from a bacterium found in the soil, it worked. It was the first real cure, a miracle pulled straight out of the dirt, saving who knows how many lives, and winning Waksman a Nobel Prize. So Waksman's work really opened the floodgates. But that hunt? It is far from over. Today, we are still digging into that massive, untapped potential in the soil. The big difference is now we have technology that Waksman could have only dreamed about. The big, game-changing idea today is something called the metagenome. See, the huge problem we've always had is that we can only get about 1% of the microbes from the soil to actually grow in a lab dish. The other 99%, we couldn't study them. But metagenomics lets us skip that step entirely. Scientists can now just pull all the DNA straight out of a soil sample. It's like instead of trying to interview every single resident of that microscopic city, we just download the entire city's library, getting the blueprints for everyone, including that mysterious 99%. What this does is give us access to this unbelievable genetic library written by nature over billions of years. We can scan through it, looking for the specific genes that make new antibiotics or powerful enzymes we've never seen before. We're not just looking for the little chemists anymore. We're reading their secret recipe books to find the next wave of life-saving treasures. And that really leaves us with this one big question. I mean, from figuring out how to grow our food to finding cures for the world's worst diseases, this tiny world under the ground has, again and again, completely changed our own. So seriously, the next time you're just walking outside, think about that bustling, invisible city right under your shoes, and just wonder, what other secrets are still down there, just waiting for us to find them?